Good morning students. Today our lecture will be on structure of sperm and fertilization of egg. This is chapter 3, human reproduction, and we are going to understand this thing. Now you know that gamete, male gamete is sperm and female gamete is egg. So site of sperm production, where are sperm produced in human? So you can see an image of section of seminiferous tubule. So this is the outermost layer, tunica albuginea, and on this there is basement membrane inside to it. So outer layer is having Leydig cells, connective tissues, and inner layer of the seminiferous tubules are having layer of cells. So this is the first layer of cell, which is spermatogonia or sperm mother cell. After that, the second layer is made of, or we can say sperm mother cell has grown in size, growth has occurred and it becomes primary spermatocyte. Then primary spermatocyte is undergoing meiosis 1 to form two cells which are known as secondary spermatocytes. Now secondary spermatocytes again form one more meiosis division to form four cells called as spermatids. So these spermatids, as you can see, are circular in shape, but later on when they fuse their head in the Sertoli cells, then they are going to mature and make the flagella out of it. So now you can see in the next diagram that this is the spermatid that is circular in shape, having nucleus, having Golgi body and lysosomes, and it is maturing in the next figure as in a torpedo stage, like triangular head-like structure, having centriole in the center and middle piece here as the sperm. So this is called as maturation of sperm. Now coming to the next, this is the sperm structure. It is having three main structures. You can see the top is the head, then in the center there is small neck, and then a propulsion system means a system which will help it in movement, generating force to move. And then lastly there is a tail or flagellum. There is also a sac at the top, a cap-like structure that is having enzymes that enables to fuse sperm nucleus to fuse into the egg. Now coming to the sperm head. What does sperm head contain? Sperm head contain condensed haploid nucleus and a cap-like structure called acrosome. So cap of the sperm has in digesting enzymes called as, that cap structure is known as acrosome. The acrosome is modified secretory vesicle derived from the Golgi apparatus. So Golgi apparatus in the spermatid cell has only changed into the acrosome. So it is having enzyme called hyaluronidase that digests outer layer of egg cell. Now coming to neck and middle piece. So neck has major motor portion of the flagella of the axoneme, means here there is a centriole, network of centriole by which microtubules in 9+2 arrangement will arise, and they will be completely extending to the flagellum or the tail portion of the sperm. Now middle piece. Middle piece consists of one mitochondria on each of the side, and so a bunch of mitochondria is present in the middle piece of the axoneme, and then this generates the force for the sperm motility and provided by the hydrolysis of ATP produced by mitochondria. So mitochondria, as you know, is the powerhouse of it. There are large number of mitochondria present in the middle piece of the sperm. Now coming to sperm activation. The ejaculated sperms are able to move, but the capacity to bind the egg and fertilize it will only come after certain chemical reactions that take place in the female reproductive tract. So here you can see a human sperm is there, but it gains maturity in the epididymis within the male reproductive tract, but it gains capacity to fertilize only in the female reproductive tract. So the location of this process is known as capsitation means attaining the ability to fertilize, capacity to fertilize. Now, there are two videos which I want you to see to understand fertilization more quickly. I think some problem is there in the video. This video is not playing, but it's okay. I will share the link of the video in the below the comment section. You can play from there, or I will message this link later on, but we will proceed further. So, steps of fertilization. Sperm and zona pellucida binding. This is the first step of fertilization. Binding of sperm to the zona pellucida is a receptor-ligand interaction with a high degree of species specificity. Means every particular species sperm can only bind to its own species egg. For example, the sea urchin sperm will only bind to the sea urchin egg. And there is incompatibility between sperm and egg of two different species. So the carbohydrate groups of the zona pellucida glycoprotein function as sperm receptors. There is a lock and key mechanism. So human or single species sperm can only fertilize the egg of that species. The sperm molecule that binds this receptor is known with certainty, and indeed there may be several proteins involved in this fertilization or binding. Now, after this, there is acrosomal reaction and sperm binding. So, acrosome. Now, the cap structure of the sperm is having acrosome. So, here the acrosomal reaction will occur. Binding of the sperm to zona pellucida is easy part of fertilization. The sperm then faces the daunting task of penetrating the zona pellucida to get to the oocyte. So, binding to the zona pellucida or outer layer is very easy, but to cross that zona pellucida layer is little difficult. So, acrosome releases huge amount of enzymes through the lysosomes, and then it digests the zona pellucida, then it penetrates and reaches further in the egg. The acrosome reaction provides the sperm with an enzymatic drill. It causes a drill through the zona pellucida, and then zona pellucida, some of the proteins serve as sperm receptors, stimulate the series of events so that the fusion of both the cells fuse. Sperm that lose their acrosomes before encountering the oocyte are unable to fertilize. So, if acrosome integrity is very important, and some of the ejaculated sperm, if not having this acrosomal integrity, they cannot fertilize the egg. So that is the problem. Sometimes this is the cause of male infertility. Now, penetration of zona pellucida. The constant propulsive force of the sperm flagellating tail in combination with acrosomal enzymes means both things are necessary. The constant force of the middle piece of the sperm, propulsive force, and the tail of making the sperm to go ahead, and the enzymes which are released from the acrosome head. Both are responsible to allow the sperm to go and penetrate the zona pellucida. Now, next, sperm oocyte binding. Now, if it has crossed the zona pellucida, now both the cells will bind. Once the sperm penetrates the zona pellucida, it binds and fuses with the plasma membrane of oocyte. Binding occurs at the posterior acrosomal region in the sperm head. Means now both the membranes, cell membranes will bind, and egg activation will occur now. So, egg activation is also known as cortical reactions. few calcium or cortical granules will be released from the egg membrane to outside, and there will be a lot of metabolic and physical changes called as egg activation will occur, and it will include rise in intracellular concentration of calcium, completion of the second meiotic division, so called cortical reaction. Means inside the egg there is large amount of calcium will come inside. The egg cell will be permeable to calcium ions, and therefore the cortical reaction and the second meiotic division will take place. Now the zona reaction. Zona reaction refers to an alteration in the structure of the zona pellucida catalyzed by the proteases of cortical granules. So cortical granules which are ejected out from the plasma membrane of egg will have certain enzyme, protein digesting enzymes. So they will be doing some of the reactions to chemical reactions in next 10 seconds or few seconds, so that polyspermy is avoided. Means if first sperm has fused the egg, now no any other sperm can fuse it. So this is called as block to the polyspermy. The zona pellucida hardens and crudely put this analogous to setting of concrete. Means now the cell will be covered by a concrete hard tough structure, so that no more another sperm can fuse with it. And sperm receptor in zona pellucidas are destroyed. Now no more receptor is present to accept any other sperm coming. Now post-fertilization event. Now the fertilization afterwards, what happens? Following the fusion of fertilizing sperm with the oocyte, the sperm head is incorporated into egg cytoplasm. Now the sperm head is gone into the egg cytoplasm, and the nuclear envelope of sperm disperse. The chromatin material, which was highly condensed, get to loosen up, and this process is known as decondensation. Now the chromatin material will decondense, and mitochondria are degraded rather than incorporated. The sperm mitochondria are degraded. So only what is gone inside the egg is sperm chromatin material, genetic material is coming from the sperm. Chromatin from both the sperm and the egg are soon encapsulated in a nuclear membrane, forming pronuclei. So both of the haploid chromatin materials from egg as well as from the sperm are packed into single nuclear membrane now, and this is known as pronuclei. Now then afterwards, each pronucleus contain a haploid genome. They migrate together, their plasma membrane break down, and two genomes condense into chromosomes, thereby reconstituting a diploid organism. Now both the pronuclei will come together to make a diploid chromosome. Both arrange together as single diploid nucleus, and the centrioles of the sperm only will arrange those both of the chromatin material at the equatorial or at the plane, means at the same position. So with this, the fertilization and the sperm structure and the egg structure, this all I hope is clear to you, and I will be mentioning the link of the YouTube video in the comment section. Thank you.